So for today's disease that you're going to look at is malaria. So the learning outcome for malaria is state the name and type of causative organism of malaria, explain how malaria is transmitted from an infected to an uninfected person, also discuss the roles of social, economic, and biological factors in prevention and control of malaria. And actually, this one is no longer in your syllabus. Okay, this is no longer in your syllabus. Discuss the factors that influence global patterns of distribution of malaria and assess the importance of this disease worldwide. Okay. So malaria is an infectious disease of human as well as other animals. That means the pathogen can infect our body and also can infect animals like monkeys. Okay, so it is caused by a protoctist. So you know that protoctist is actually a kingdom of organism that is made up of eukaryotic cell. Okay, and they are unicellular organism. Okay, so protoctist. So genus Plasmodium. So of the genus Plasmodium. Now transmitted by what? Transmitted by vector, or you can call it the disease vector. Okay, a vector means is a medium. A medium. So this vector, which is a mosquito, it is a medium that will carry the pathogen from one person who is infected to an infected person. So the vector is a medium, a transport medium. So this vector, which is a mosquito, will carry the pathogen from infected person and then transmit it to an uninfected person. So it is a vector, a disease vector. Or it can carry from an animal also can. Okay, can carry from animal and then transmit it to an uninfected person also can. Okay, so this is a vector. Vector is an organism that can transfer the pathogen from a host cell, the infected person, to an uninfected person, or from the animal to an uninfected person. Okay, so protoctist. So what is protoctist? So it is a group of simple, mostly unicellular eukaryotic organisms that do not fall easily into either fungi, plantae, or Animalia kingdom. So this is the definition of protoctist. It's a kingdom of eukaryotic organisms which are mostly unicellular, and it's very difficult to classify them into the kingdom of plantae, animalia, and fungi. So that's why they are all classified under this kingdom, protoctista, like amoeba, paramecium are all under protoctista. Okay, because they're all unicellular eukaryotic organism. Okay, vector is an organism which carries the pathogen from an infected person or animal to an uninfected person. So you need to learn up the definition of those terms. Okay. So the vector for malaria is a female Anopheles mosquito. Now you need to write female because only female will suck blood. The male will not suck blood. Okay, the male will feed on plant juice. Okay, why the female need to suck blood? Because they need the protein from the blood in order to make eggs, to produce their own eggs. So that's why they have to suck the blood. So only female Anopheles mosquito can become the vector for malaria. So there are 4 types of Plasmodium. So you've got Plasmodium falciparum. So when you write Plasmodium falciparum, you need to underline the P, you need to underline the falciparum, and make sure that there's a single spacing between the P, the dot, and the F. Okay, between the dot, there's an empty space here, one space, followed by falciparum. Okay, so we have got Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, and Plasmodium malariae. So out of these 4 types of species, the one that can cause deadly disease for malaria is Plasmodium falciparum. This is the one that can cause deadly disease, so therefore it causes majority of the death from malaria. So Plasmodium falciparum, if you look over here, what you can see is it will go it will go through different stages of life cycle. So at different stages, the shape of this cell, because it's unicellular organism, so the shape of the cell will be different. Okay, so if you look at this here, this is the stage where it is ring shaped and it grows inside the red blood cells. Okay, it grows inside, so it multiplies inside the red blood cells. And then slowly, it becomes spherical in shape. Okay, and after that, it becomes elongated. So at different stage, it's got different shape. So in order to diagnose a person with malaria, usually we will take a blood sample from the person and we observe it under the microscope to look into the red blood cells, whether there are any cells which contain nucleus. Because Plasmodium is a eukaryotic cell, so eukaryotic cell has got nucleus, but red blood cell does not have nucleus. But the moment you look into red blood cell, you start to see like ring-shaped cell with nucleus or many cells with nuclei inside red blood cell. Most probably it is malaria. So this is the way we diagnose for the presence of malaria in a person. Okay. So what are the symptoms of malaria? Is fever, headache, muscle pain, chills means feeling cold, nausea means wanting to vomit, shivering, sweating, enlarged spleen and anemia. Anemia is because once our red blood cells are invaded by the Plasmodium, it will start to divide inside the red blood cell, and after that, the red blood cell will burst to release all the newly formed Plasmodium cells. So therefore, a lot of red blood cells will eventually burst, so this person will got lack of red blood cells, so that's why anemia. Okay, and then Plasmodium falciparum causes more severe symptoms like very high fever, convulsion means the muscles will contract involuntarily. Okay, you will shiver here, there, everywhere without your control. Okay, and then coma means a person will become unconscious already, will fall into coma means unconscious, and which can lead to death. Okay. So because Plasmodium is able to infect red blood cell of all ages, so it can infect red blood cell of all ages, means whether it's 1 month, 2 months, 3 months, or red cell, you infect all the red blood cells, and then the infected red blood cell becomes very sticky. So when they become very sticky, they stick to the blood vessel and they cannot move. So therefore, it will block the flow of the blood to the targeted organs of the blood vessel. So when the blood vessels has got red blood cell that's sticky, right, and then it will not flow, they stick together, so there'll be no blood flow. So imagine if this is carrying the blood to your heart or to your brain, there'll be no oxygen supply. There'll be no glucose supply. So the cell cannot carry out aerobic respiration, no ATP. So the cell eventually will die. So it's very serious. That's why this person can die. Okay, because of Plasmodium falciparum infection. It caused the red blood cell to become sticky. Okay, so these are the signs and symptoms of a person who has malaria, which I've mentioned already. Okay, so diagnosis of malaria, as you can see here, this is a blood smear, and inside the red blood cell, you can see like ring-shaped cell with nucleus, right? And then these are the malaria cells that have been released from the red blood cells already. So this is another stage. And this is malaria cells that are still inside a red blood cell. You can see that the entire red blood cell is fully filled with malaria, right? And this is another stage, right? This is another stage here. Red blood cell. Okay, this is very interesting. Now, this red blood cell is red blood cell of other animals. It is not red blood cell of human. Okay, now red blood cell of certain animals, they have got nucleus one. Can you see that? All of them got one nucleus one. And then this is the Plasmodium cell in this red blood cell. Okay. Yeah, so therefore, there are in the past year where students are given before the slide of red blood cell, but very interestingly, all the red blood cells contain nucleus, and students need to actually draw the detailed drawing of the red blood cell and label it. So whatever you see, you need to draw, because there are exceptions. Exception means something that's different from human red blood cell that you've seen, but you already know how to identify nucleus. So therefore, when you look at it, you look at the shape of the cell. The shape of this red blood cell is not biconcave shape. It's more like oval shape. So different organisms, they got red blood cell, but the shape and the content could be different from human red blood cell. So this is something that you need to know how to identify by looking at the slide itself. Okay, so diagnosis of malaria, we have got microscopic examination of the blood for the presence of the Plasmodium, which is actually unicellular and is got nucleus, is nucleated cell within our red blood cells. Okay, and of course, the second method is a dipstick test. Dipstick test is a rapid test for Plasmodium falciparum antigens in the blood without the need for laboratory. That means to say, if we don't have the lab, right, we don't have the microscope facilities, and we don't have the specialists who know how to use a microscope and know how to make the slide and know how to view it and identify and diagnose the Plasmodium, then we need to do dipstick test. I mean, dipstick test means you take the dipstick and then you put a drop of blood onto it, and after some time you see one line, it means what? Two lines means what? Just like the pregnancy test. Okay, one line means not pregnant, two lines means pregnant. Okay, so this is testing for the presence of the antigen of the Plasmodium, because every cell has got antigen on the surface, which is a name tag of the cell. So therefore, you use a dipstick to test for the presence of the Plasmodium antigen. So if it's positive, that means the antigen is there, that means the Plasmodium is in the blood of this person. So this person has got malaria. Okay, so this is a microscopic examination of the blood. So first you put a drop of blood, then you have to spread it out like this. So you can see that if you got a drop of blood, even the light cannot pass through, it's too thick already. So you have to spread it out with another slide to make a very thin layer that the red blood cells and the white blood cells do not overlap. So you can see individual cell very clearly. So you can see the content of the cell. Okay, so this is called the blood film, right? Blood film. Film is very thin layer, right? This is a blood drop, very thick layer. All the cells overlap, cannot see. Okay, right, next. Next. Explain how malaria are transmitted. So let's look at the transmission of malaria. That means how the Plasmodium can be transmitted from infected person to an uninfected person. So let us look at the transmission cycle of the Plasmodium. So we always start from an infected person first. Okay, so from an infected person, majority is female Anopheles mosquito will suck the blood from this person. So when they suck the blood, the red blood cells which contain the Plasmodium will all be sucked into the gut of the mosquito already. So it will go into the gut of the mosquito. Okay, so from the gut of the mosquito, the Plasmodium will then migrate to the salivary gland of the mosquito. Okay, and then the mosquito will be flying around, and then it will start to land on an uninfected person, and it will start to take a blood meal. Okay, I'll say, use the term called take a blood meal from this uninfected person. So in order to take a meal, it will have to first inject saliva into this person. So when the mosquito put its proboscis, right, like the needle, into this person, it will first inject its saliva, because its saliva contains this chemical called anticoagulant, right? Anticoagulant means, anti means prevent, coagulant means to coagulate means solidify. So this is a chemical that prevents the blood from clotting while the mosquito is sucking the blood. So it has to inject anticoagulant first from the saliva together with salivary gland. So when it injects the anticoagulant, then the Plasmodium that are residing in the salivary gland of the mosquito will be injected into the bloodstream of this person. So this is how the Plasmodium is actually being transferred from an infected person to an uninfected person. So inside this person now, the Plasmodium will first will first go into the liver cell, migrate into the liver cell, and then start to multiply inside the liver cell. So from one cell, it becomes many cells in the liver cell until the liver cell cannot withstand anymore, and then it will burst because the number of Plasmodium cells in the liver cell will start to increase as the cell divide from 1 to 2, 2 to 4 to 8 to 16, 32, 64, and 128, and so on. Okay, so eventually liver cells will burst to release the Plasmodium. And then the Plasmodium will then go into the bloodstream and start to infect the red blood cell. So this is the second cell that the Plasmodium will divide in our body. The first cell is the liver cell. Then is the red blood cell. So it will again multiply in the red blood cell until the red blood cell can no longer expand anymore. It will burst. Okay, and when it bursts, it will release more and more Plasmodium. So in every stage of the life cycle of the Plasmodium number will increase because every stage it will multiply. Okay, even in the mosquito also. Okay, so once the red blood cell lyses, it will release the Plasmodium, and then after that, this person, when the next mosquito come and take a blood meal, it will then suck in the Plasmodium that are found in the blood plasma as well as in the red blood cell. Okay, so this is a complete cycle of the Plasmodium. So the main pathway is through the disease vector. Disease vector, which is an organism which can pass the pathogen from one person to the next person. Okay, so disease vector is an organism. Vector, when you look at the word vector, vector's main function is it acts as a transporter or a carrier. So it's supposed to carry the pathogen from one person to another. So that's the meaning of a vector. Okay, a transporter or a carrier. Now, of course, apart from the mosquito, there are a few other ways that the Plasmodium can also be transferred from an infected person to an uninfected person. All right, so we have looked at number one, right, which is the female Anopheles mosquito. Number two is from the mother to the fetus directly through the placenta. That's possible. Number three is via blood transfusion. That means when a person donates the blood, this person has got malaria. And then if the blood is not screened, right, then this blood will be donated to another person. Then the person will get this malaria in the transfused blood. Okay. And of course, sharing of unsterile needle. Okay. That means to say that especially for drug addicts, right, they have to spend a lot of money buying the drugs. So they will actually try to reduce costs like needle. They don't have to buy a lot of needle. They keep on using needle. And sometimes they even share needle with other people. So when they share needle is when, you know, the needle is placed into the vein and inject the drugs. So when you take it out, you will still have got fresh blood there. So when they have not dried yet and another person immediately use it, then of course the blood from this person will be also introduced to another person if the person used immediately. And of course, if the blood dry off, then of course the Plasmodium will also die because the Plasmodium also dry off already. But if they share the needle and take the drug at the same time, that is when the blood from one person can be transferred to another person because the needle is contaminated. Contaminated is contaminated with Plasmodium. Okay. Of course, there's one more thing that I didn't mention here is blood-brain barrier ruptures and hemorrhage. Blood-brain barrier means inside the brain, the barrier between the blood, between the blood vessels and the brain rupture, and all the blood, it's like your blood vessels has ruptured. So all the blood will flow out from the blood vessels. So this is possible. Okay, right. Now, this is life cycle of Plasmodium. It is not in your syllabus. So what I want you to look at is only like Number one, Plasmodium, because it's a eukaryote, it has got a few stages in one complete life cycle. A few stages in one complete life cycle. And in each stage, the shape of the cell will look different. Okay, so therefore, when the mosquito first inject the Plasmodium from its salivary gland, so Plasmodium that reside in the salivary gland, which goes into the bloodstream of this person, the Plasmodium exists in the form of spiral shape. Spiral shape is like snake like that, okay, spiral shape. And then it will then go into the liver cell and start to multiply in liver cell until the liver cell cannot expand anymore. It will burst to release many Plasmodium. And now you can see this Plasmodium, the shape is different already, right? Because it's a different stage of life cycle. And then this Plasmodium will then infect the red blood cell and start to multiply in the red blood cell. So this is when you will see the ring shape, okay? The ring shape. And then after that, when it's released, it slowly becomes a gamete, okay? So you got a male and female gamete that will suck in by the mosquito and go into the gut. And then this male and female gamete will then fuse together. And when they fuse together, they will migrate into the salivary gland. And then the next cycle will continue when the mosquito take the next blood meal. So this is a complete life cycle that you don't have to memorize any of the name. You just have to know that each stage has got a different shape. You don't have to memorize the shape also. Okay, you don't have to memorize the shape, but just to show you that there are a few stages of life cycle for Plasmodium because it is a eukaryote, so it's more complex. Okay, right. So next, so there are a few videos that are very, very interesting about Plasmodium life cycle. So let me show you the video now.
The malaria parasite.
So this is a very important section of every infectious disease that you're going to learn. That means you need to discuss the roles of social, economic, and biological factors in the prevention and control of malaria. That means when we want to prevent or control this disease, we need to focus on the society, what the people can do. We need to focus on the economy. Do we have enough money to provide this kind of preventive as well as control measures to our society? Biological factors mean we must consider the pathogen. How does the pathogen is being transmitted from a person to another person? So we need to know how it's being transmitted, and therefore we know how to break the transmission cycle. So these are the three factors that we need to consider every time when we want to discuss about how to prevent and control disease. So you already learned in the previous disease that the differences between prevent and control, right? Right? Because prevent means when a person is not infected by disease yet, so it's uninfected person, you need to prevent this person. So prevention is here. Prevent this person from getting the pathogen into the body. So what are the actions that you're going to do here to prevent this person from getting the disease? So this is called prevention, isn't it? Okay. And what about control? Control is you are focusing on a person who already got the pathogen in it. So the infected person. Infected person. Or even the mosquito, which is the vector, which is already carrying the pathogen in it. So you want to control them so that they will not spread the pathogen to other people. So when you say control, you are controlling the spread of the person by controlling the infected person as well as the vector. So over here, you can see that we are focusing on three things here in order to prevent and control. To prevent, we need to focus on the To prevent, we need to focus on the uninfected person, what this person can do, right? if you have not got COVID, for example, what must you do to prevent yourself from being infected by COVID virus? So you are the one, we are the one uninfected person should be preventing. What action to take? Like for us, for COVID, it's wearing masks and so on, right? So what about for this disease, right? And also for the vector, because the vector is carrying pathogen. So what can we do to the vector to prevent the vector from passing the pathogen to an uninfected person, or to pass the pathogen from infected to uninfected person? So how to actually stop this step here, stop this step here, stop this step here. Okay, so therefore this part here is prevention. Prevention, right? This part here is prevention. And this part here is control. Control the person. You can also say control the vector. When you control the vector, you are preventing the vector from coming into contact with this uninfected person. So control and prevention can overlap in this case because we're trying to control the mosquitoes. So we can control the mosquitoes by controlling their number. So we try to reduce their number. We do not allow their number to increase. We try to remove them if possible. Okay, so these are the things that we need to focus on in prevention and control. So let's look at the detail, prevention and control. So prevention and control can be done in 3 entry points within the transmission cycle, because every time you say prevention and control, always have got the transmission cycle in your mind. Where can I break the cycle? Where can I break the cycle, and how do I break the cycle at every point in my transmission cycle? So control in human. So for uninfected person like us, we try to avoid being bitten by the Anopheles mosquito, female Anopheles mosquito. Okay, and then control of mosquitoes. So we control human, right? Okay, we control ourselves, right? So prevent ourselves from being bitten by mosquitoes. And number 2 is control of mosquitoes. So we try to do something to reduce in the mosquito numbers. We do not allow them to breed, so the number will not increase. And then, of course, the third one is we try to control the Plasmodium. So control human, control mosquitoes, control the Plasmodium. So we prevent Plasmodium from infecting people. So there are 3 focus that we can control: human, the mosquito, and the Plasmodium. So in human, what we can do is try to avoid being bitten by the mosquitoes. Okay, so when we go out to the wild, we have to, where there are a lot of mosquitoes, try to use insect repellents, roll it on your skin or spray it onto your skin that's exposed, right? Or else cover your skin. So wear long sleeve, wear long pants, okay, to cover your skin with cloth so that they will not bite you. And wear something that the fabric is thicker, not the very thin fabric, and then the mosquito can still poke the proboscis through the fabric. Okay, sleep under the mosquito nets. Impregnated with insects means spray the mosquito net with insecticides. Okay, so this is control in human. What about control of mosquitoes? Yeah, we want to control them so that the number will not increase, right? So that means you try to spray insecticide and oil over the water surface. So when you got the oil, right, it will prevent mosquito larvae and pupae from breathing. So they cannot get oxygen, they will die due to lack of oxygen. And of course, insecticide will kill those larvae and pupae. So you can spray insecticide and oil over water surfaces around your house and a nearby area. Then drain any stagnant water in marshes and ponds. When you got water start collecting right after rain, try to drain them out so that it will not allow mosquito to breed. Okay. Clear vegetation to prevent water collecting. That means when you got a lot of plants around that the water can collect below the plant and so on, we try to remove all this unused vegetation, okay, or extra vegetation that causes the water to start to collect. Okay, biological control. Biological control means we use an organism to control another organism. That's why biological control. I use a biology means I use an organism to control the Plasmodium. So what do? I can rear fish in the pond, so the fish will feed on the mosquito larvae. I can also spray Bacillus thuringiensis, which is a type of bacteria which can produce Bt toxin. Okay, so Bacillus thuringiensis is a bacteria that can produce Bt toxin. So the word Bt stands for Bacillus thuringiensis. So when you spray this bacteria into the water, like in the pond, in the lake, okay, it will start to produce Bt toxin, and this Bt toxin can kill the mosquito larvae, but it will not be toxic to other aquatic organisms like fish inside the pond. Okay, so these are measures to control the number of mosquitoes. And thirdly, how to control the Plasmodium. Use anti-malarial drugs. Okay, use anti-malarial drugs. So these are anti-malarial drugs on the right side, right? So number 1 to number 4. So number 1 to number 4, we've got quinine, chloroquine, mifeprine, and proguanil. So these are the 4 types of anti-malarial drugs. So number 1 to number 4, that means all of them are used for treatment. Treatment means when a person is already infected. Like just now you see the video. The moment this patient is put on drip, where this any one of these anti-malarial drug is being transferred into the body, then it will start to kill the pathogen. So this is for treatment purpose. Okay, so number 1, A is for treatment purpose. B is they will be used as prophylactics. Now, prophylactics stands for preventive drugs. Okay, preventive drugs. That means you take it before you are being infected. For example, you know that you're going to travel to an area where there is endemic with malaria. Endemic. Now, endemic means it's describing a disease that exists all the time in a small number, in a small area. Okay, so when you want to travel to those countries which is endemic with malaria, then your doctor will advise you to take the anti-malarial drugs as prophylactics, means preventive drugs, so that when you arrive there and when you are infected with the Plasmodium, your body already got a certain concentration of this drug to quickly kill them, so that you prevent them from multiplying in your body and start to destroy our liver cells and our red blood cells. So then these anti-malarial drugs will be called the prophylactics when you use it as preventive drugs. But when you use it for treatment, it is called the anti-malarial drugs. Okay, so they've got both effect, right? Kill before enter or kill after enter, right? Kill before pathogen enter or kill after the pathogen enter. So there's this another drug called antibiotic doxycycline, which is also used for treatment. So this is the only antibiotic. Now the rest of them are not antibiotics, but they are drugs that are used for treatment. So they're called anti-malarial drugs. So later on, you will learn what is the meaning of antibiotics in another session, right? Not all drugs are antibiotic. Antibiotic is only one type of the drugs that are used to treat infectious disease. Only one type of all the rest of the drugs that we use. Okay. Now, so I've already elaborated this part already. Now, one more thing we have to be very careful is every time you use any chemical at all, it can cause resistant strain to start to develop. Any kind of chemicals. If you use insecticide, you keep on spraying the same brand of insecticide. After a long time, you start to see that, how come the mosquitoes like no effect already, because they become resistant to that insecticide that we use. At the same thing, if you use the drugs for a long time, then after some time, the Plasmodium will also develop resistance to that drug, and the drug is no longer effective. So that's why we have to keep on changing the drugs that we use to treat the same type of disease, because drug-resistant strain of the Plasmodium can grow, and then insecticide-resistant strain of the mosquito can also develop. So every time we use chemical, this will happen. Okay, so this is in the new textbook, which says that currently the most effective treatment against malaria, because of the drug resistance, right? They keep on finding new ways to treat. So lately, the most effective way of treatment is called artemisinin-based combination therapy. So artemisinin, ACT, okay? Artemisinin-based combination therapy means they take the chemical that is derived from the plant called Artemisia. So that's why the name artemisinin, okay? Combined it with the normal anti-malaria drug that we use. Okay, the latest effective one is mifequine. So they combine the chemical obtained from Artemisia plant with the mifequine to be used as a treatment, as a therapy. Okay, and this is very effective against the deadly Plasmodium, which is Plasmodium falciparum. Yes, you are right. Also in the new textbooks, this is what I want to show you now, because there are a lot of new things happening. So in the new textbook, okay, so in the new textbook, it is said that there's a new vaccine, right? RTSSAS01, short form RTSS, known as Mosquirix. Okay, and it's very interestingly, it is an injectable vaccine. So usually we inject vaccine in the body, right? It provides partial protection only against malaria in young children. So basically it's for young children, because I think young children, if they are not protected under the mosquito net, right? So let's say the house is open and mosquito can fly in anytime. Then how do we protect them? So we have to provide them with vaccine, right? At least can provide partial protection. So this one is developed in November 2016, recently, a few years back only, right? So WHO announced that this vaccine would be used in pilot projects. Pilot project means this is a first trial, in a big scale first trial. After this has been developed in the lab, the first testing, okay, the pilot projects. So they have selected areas, right, in 3 countries. Of course, it must be the countries where have got very serious cases of malaria, right? So usually it's in Africa region. The reason is in Africa region because it's this region, the temperature is very suitable for Anopheles mosquito. So Anopheles mosquito can live very well and it can live very long. So when they live long enough, it allows the gametes that have entered their digestive tract, right, the gut, can start to fuse together, and then they can have time to migrate to the salivary gland of the mosquito. And therefore, it can be injected to another person, because the Plasmodium, the Anopheles mosquito is the one where it can survive very long, to allow enough time for the Plasmodium to start to change from one stage of life cycle to another. So from the gut into the salivary gland. Okay, so it's in this country, Ghana, Kenya, Malawi, right, all in the Africa. So they are given this vaccine to try out and see how effective it is. So funding was secured for the initial phase of the program, and vaccines began in Malawi in early 2019 only, like 2 years ago, right? 2 years ago, it started in Malawi. And other vaccines are under development or being tried. It means still being under developed in the lab. Being tried means already being tried by people, right? You can find out the progress in the developing vaccines to protect people against malaria by searching online. So because it's very new, so we need to actually search it online, okay, for the latest development. So, of course, there are always research and development. Will always try to do a research to improve on the way to prevent and control the disease. Okay, so what are the factors that may lead to improvement in the control of malaria? So use of modern techniques, right? use of modern techniques like gene sequencing and drug design to improve on the drugs, especially vaccine. You'll know why already later on, because vaccine, later on you'll learn, okay, never mind. Development of vaccines targeted against different stages of parasites. So we know that the parasite got different stage, right? So we can develop vaccine that can target against different stages of the parasite's life cycle, so that whichever stage the parasite in, there will be vaccine that can act against it. So therefore, it will be effective for us, right? Regardless of whether it is still, it's just being injected to our body, or already travel between liver and red blood cell after liver cell already, waiting to be sucked in by another mosquitoes. So it's very important to have that. Okay. And then we have got the last point here, which is a renewed international will to remove the burden of malaria from poorest parts of the world and lead to generous donations from wealthy individuals and foundations. So of course, if you want to provide the vaccines and as well as the anti-malaria drugs and education to the people, of course, in the poor country where the malaria is still endemic, we need funding. So we need to obtain donations from large foundations, from the governments and from individuals to fund the WHO to provide the medical resources like the vaccines, the drugs, the medical staff, okay, to administer these drugs, to provide diagnosis for the people in the poor country, so that at least we can eradicate this disease totally from the world, right? From the world. So that's what we are working on, especially WHO, working on removing infectious disease as much as possible. So let's recap the malaria. So malaria is caused by the genus Plasmodium. There are Plasmodium falciparum, vivax, ovale, and malaria. So Plasmodium falciparum is the most serious one, which can cause deadly disease. Method of transmission is by the insect vector, which is female Anopheles mosquito. When you write the word Anopheles, you must underline because it's a genus name and you must start with capital letter. Global distribution is usually in the tropics and subtropics region where the female Anopheles mosquito thrive. Okay, can survive that. Incubation period is from a week to a year. Wow, 1 year. Okay, site of action of pathogen is in liver, red blood cell, as well as in the brain. Clinical features we already mentioned: fever, anemia, nausea, headaches, muscle pain, shivering, sweating, and enlarged spleen. Method of diagnosis is microscopic examination of blood as well as dipstick test for the malaria antigens in the blood. Annual incidence is quite high, 207 million cases of malaria in 2012. About 80% are in Africa. Annual mortality rate also high, death rate 630,000 number of deaths in 2012. And about 90% of deaths are reported from Africa. So that's why WHO is making the pilot project in Africa to help those people. So that's all for today.